Hello everyone and welcome to Instant Biology by Dr. Nilab. In today's lecture, we would be discussing about termination process in DNA replication. So if you have been following my YouTube videos, you would realize that I have already talked about initiation process, I have already talked about elongation process, and this would be the final video on termination. Now just a quick reminder that we have already made videos in Hindi about initiation, elongation and termination. So if you are more comfortable in Hindi, then you can switch over to those videos. Otherwise you can stick to this video. This would be in English. So let us come back to what we have to discuss today. We are talking about DNA replication, and in that we would be talking about termination. So what was DNA replication? Just a quick reminder. In DNA replication, we said that one DNA strand has to be copied completely into another DNA strand, and that would be complementary to the first strand. And in this way, what we are doing is that we are making continuous copies of the DNA. This process is called as DNA replication. In DNA initiation process, what happened is we started with the replication. That means all the protein machinery was assembled and everything was being done. In the elongation process, what was done is nucleotides were continuously added onto the growing chain. In this way, we were synthesizing the chain one by one, adding one nucleotide after another. In this way, we were synthesizing the chain. Now, what we are aiming at is terminating the DNA replication process. Now, what does termination means when we are talking about prokaryotes? Remember, in this particular lecture, we are aiming to discuss DNA replication in prokaryotes. So, in prokaryotes, you might remember that the DNA strand is circular in nature. So, suppose this is the DNA strand, it is circular in nature. Now, when another copy of this DNA strand is made, so this was circular, when another copy of the DNA strand is made, it appears to be like this, or it comes into this shape like this. Now, both the DNA strands, one that has been made newly or recently, and the other one that is old, now these two strands are joined together in a concatenated fashion. That means they are connected together like this. Now, in termination, what happens is we need to break this concatenation and release both the strands separately. This is what needs to be done in the termination process of DNA replication. So this is how we would be talking about step-by-step process. We would be understanding each step in termination, how it is done, and the different enzymes that would be required. So let us start. So DNA replication termination in prokaryotes, first of all, the first thing that has been written over here is, that is very important, is bacterial genome. In bacterial genome, the replication is bidirectional from ORIC. Now you would remember ORIC is the region from where the replication starts in both the directions. Because I have already said that the replication is bidirectional in case of prokaryotes. So suppose this is the ORIC, from this over here also the replication will proceed, and over here also the replication will proceed. So this is what I mean by bidirectional mode of replication in case of bacterial genomes. Now the two replication forks meet diametrically opposite to ORIC. What does this mean? Now suppose this is the point where we are starting the replication, isn't it? This was the ORIC. Now the replication fork, one replication fork would be moving like this, other replication fork would be moving like this. Both the replication fork would be moving like this, and they would be meeting each other at a point just opposite, diametrically opposite to the ORIC point. So suppose this was the ORIC point, opposite to the ORIC point, this is the point. So both the replication forks would be moving and they would be meeting opposite to the ORIC. This is what I want to say. So this is the second point that has been written or that has been mentioned on your screen. Now this region, the region that I am talking about over here, the region that is opposite to the ORIC, that the region that is present over here, is called as the terminus region. But naturally, it should be definitely called as the terminus region because the termination is going to be ending over here only. So just diametrically opposite to the ORIC, the termination needs to be ended, the termination will end, and this region is called as the terminus region. Now, what is the salient features or what are the characteristics of the terminus region? Let us understand this. It contains multiple copies of 23 base pair sequences called as ter sequences, and 7 have been identified in E. coli. What do you understand by this? Now there are 23 base pair sequences, multiple copies of them. Let me just show it to you. Let me just draw it. So if I if I draw it, so there are, let us say that, just give me a minute. Yeah, so if I draw it, suppose this was the Ori C, and opposite to Ori C is this region. This is called as the terminus region. Terminus region. Now if I zoom this terminus region, then what I am trying to say is there would be multiple repeats of 23 base pair sequences. So like this would be one repeat, this would be another repeat, this would be another repeat. So this would be 23 base pair, this would be again 23 base pair, this would be again 23 base pair. So 7 of them have been identified in E. coli. So these 23 base pair repeats are present in the ter region or terminus region. Okay, so these are called as ter sequences. Now the name comes from the terminus region only. So because they are present in the terminus region, therefore these sequences, these repeated sequences are called as ter sequences. So I believe you would have understood at least up till over here. Now let us move on to the next point. Now these ter regions that are present in the terminus, these ter sequences that are present in the terminus region, they serve as the binding site for a DNA binding protein called as the TUS protein. What I want to say is, now there is a protein called as TUS protein, TUS. Okay, and what does this TUS protein do? This TUS protein is a DNA binding protein. What does DNA binding protein mean? DNA binding protein means that it would be binding to the DNA, right? And where will it bind on the DNA? It will bind on the DNA at the ter sequences present at the terminus region. So suppose this is the these are the terminus, this is the terminus region, and over here are the let me show you, over here are the ter regions or ter sequences. Now the TUS protein will come and bind on the ter sequences. Over here it will come and bind. So over here a protein now would be binding. Let me show it to you. Suppose this is the protein, this is the TUS protein, it will be binding over here. Okay, now I believe this would be clear to you. This is the start of termination. Let us move on to the next slide. So yes, so this is the next slide. Let me briefly tell you what is TUS protein. What is the full form of TUS protein? So by TUS protein we mean that terminus utilizing substance. Terminus utilizing substance. Why are you calling it terminus utilizing substance? We are calling it by this name because TUS, T for terminus, U for utilizing, and S for substance. That is why we are calling it as TUS, terminus utilizing substance. Okay, now this allows the passage of replication fork in one direction but not in the opposite direction. What I mean to say is, this TUS protein, suppose this again I am making this, now this on the ter regions or on the ter sites my protein has been binded. Which protein? The TUS protein has binded. Now this TUS protein will allow the replication fork to move in one direction. Now suppose this is the replication fork that is moving like this, like this, like this, but it will not be able to move further. That means on one side, suppose this is moving like this, one side it will allow and the other side it will stop. This is what I want to mention, this is what I want to say. So this is the main feature of the TUS protein. I would be telling you about the details of the TUS protein also. I would be showing you a diagrammatic representation also to make it much more clear. So I believe you would have understood up till here that it allows the passage of replication fork in one direction but not in the opposite direction. It will move like this, but if it wants, it cannot move in the opposite direction. This is what I want to say. Now actually DNA B, it is also called as helicase. DNA B helicase is stopped. Why? Because you know that in the process of replication, the first thing that happens is helicase moves, right? just helicase moves before, helicase moves forward, and then following it the polymerase moves. So if you stop the movement of the helicase, then automatically the replication will stop, isn't it? So DNA B, if you remember, we have already said that DNA B functions as a helicase. Now if you are stopping the helicase, which protein is stopping the helicase? This TUS protein is stopping the helicase. Helicase is not able to move forward. Now TUS protein has stopped helicase. Now if it has stopped, then what will happen? Ultimately the polymerase will not be able to move forward, and this would be the end of the termination. This is what we are looking at in the future slides. Okay, so I believe you would have understood. So actually this is what I have written. Actually DNA B helicase is stopped in one direction and it is allowed in the other direction. We have discussed this. So both replication forks, now both replication forks, one that is coming from here, one that is coming from here, they become trapped in a very small region. I would be showing you this with the help of a diagram. So both replication forks, they become trapped within a very short region. Okay, so it ensures that termination occurs at almost the same position each time. Now let me show you a diagrammatic representation which will help you understand this. Now look at this. So this is the fork movement. If you see, this is the fork movement over here, this is the fork movement over here. There are two replication forks, mind it. Now you can see that over here color representation has been given. So this is the blue color and this one is the red color. Understand that wherever blue color is present first, it means that it is permissive. That means it will allow the DNA B movement. But whenever the red color is present first, it will not allow the movement of the DNA B or helicase. Now look at this. So when the replication fork is moving, let me show you with the help of a black pen. When the replication fork is moving, suppose in this direction, let me just, yes. So what will happen? So this is how the replication fork would be moving. Firstly, blue colored representation is there. That means this is permissive. Again, this is permissive. Again, this is permissive. It is moving forward, moving forward, moving forward. Again, this is permissive. Again, this is permissive, but over here, now it cannot move forward. Why? Because there is a red colored representation. That means it is non-permissive side of TUR. Similarly, if you move from here, blue is present, blue is present, blue is present, blue is present, but again, red is present. That means it will be stopped. So it will be stopped somewhere over here. At the same position each time when the replication is being made, DNA B helicase would be stopping for both the replication forks at a very small distance. This ensures that you are ending replication at the same point each time. I believe you would have understood this with the help of a diagrammatic representation. Now these are the two replication forks. Let me just change this. So these are the two replication forks that are being formed. Now because of the replication taking place and prokaryotic DNA in a circular format, you can see that these are the two replication forks that would be present, and they would be separating out, and they would be finally forming this kind of a structure. This is called as a concatenated structure. Anyways, so let us move forward and discuss the next slide. So in the next slide, you can see the first point is after replication, two new circles would be physically interlocked or catenated. This is what I was talking about. After the replication takes place, both the DNA strands would be fixed like this. They are in the form of, they are stuck like this. Okay, so this is called as catenated structures. What is this called as? This is called as catenated structures. Now you want to separate both of these strands, right? Because if you do not separate both of these strands, then they will not be able to work properly. So you will have to separate both of these strands. Now over here, a very important enzyme comes into being, and it has been asked in multiple examinations also. What is the name of this enzyme? So this enzyme is called as the enzyme used for decatenation process. What is the name of the enzyme? The name of the enzyme is topoisomerase 4. The name of the enzyme is topoisomerase 4. Let me just highlight it for you. This is the topoisomerase 4 enzyme, or if I classify this enzyme, then it comes under the type 2 topoisomerases. Now topoisomerases are of two types, type 1, type 2. So under type 2 topoisomerases, this topoisomerase 4 comes, which is responsible for the decatenation or removal of this two catenated structures of the DNA. Now over here you can see a diagrammatic representation also. So this was a circular DNA, then a replication forks were made, and finally this catenated structure was formed, and then topoisomerase 4 came for the help, and it separated the two DNA structures. Now this is the end of basically termination. With this, when the topoisomerase 4 comes, it removes this catenated structure. It is called as decatenation enzymes. This will mark the end of termination in the prokaryotes. So I believe you would have understood this concept pretty well. If there are any questions, you can always put in the comment section and I would be happy to solve them. So this was all from my side. If you like this video, hit the like button, subscribe to my channel, and also share it with your friends. Thank you so much. Have a very good day.